Hello everyone! Some of you guys have probably thought that you'd never see me in an online video, and others have probably seen too many of me in an online video. Well, this video was made specifically for those students who want to learn how to draw those mitosis diagrams and how to count the correct number of chromosomes, number of chromatids and DNA molecules, as well as drawing in some of the relevant structures in a cell when it's going through mitosis. And by the way, I have gone through a long journey in trying to get this video set up, and nothing in my home is working right now. My computer is breaking down. My camera's not working with me. My tablet pen isn't working with me, so nothing is working with me. But the show must go on. So I found a way to use my phone and a whiteboard and a glass table to kind of get this recording together for you guys. So let's go ahead and run over to the whiteboard and get started. All right, so here is our mitosis. I call it a virtual lab. So those of you guys who want to learn how to draw these diagrams, I strongly recommend that you draw along with me and pause the video when you need to, because this is one of the most critical things that you can do toward your understanding of mitosis. All right, so here are the learning objectives. The learning objectives here is we want to know how to draw all the phases of mitosis. We also want to draw each diagram of mitosis with the correct number of chromosomes, chromatids, and DNA molecules. And we want to be able to label all the relevant structures. Now, this video is for students who already have a basic understanding of mitosis. And so if you don't know what mitosis is and you have no clue what it's used for in the cell, I recommend that you check out other resources online or check with your instructor for more information on getting up to date with that. This tutorial and this virtual lab is just for students who want to figure out how to draw those mitosis diagrams. All right, so let's go ahead and get started first with learning how to count the number of chromosomes, chromatids, and DNA molecules inside of a cell when given. Okay, so counting DNA chromosomes and chromatids. Okay, first of all, I wanted to say that from this moment on, whenever I refer to chromosomes, I'm going to use the abbreviation CH''S. And whenever you see the words chromatids, I'm going to use the abbreviation CH''T, just to keep things short because spelling that word takes way too long. Okay, so in the diagram, you're going to see a strand of DNA. So here we go. We have here one strand of DNA. So I'm going to say 1 DNA. And by the way, I am sitting on the floor and my glass table is holding the cell phone, so it's going to look a little awkward, but bear with me, okay? All right, so we have here one DNA molecule. When we're talking chromosomes, we also have one chromosome. And the reason why I like to think as one chromosome being like one entity of DNA. And we're going to get to this concept in a little bit later. Now, chromatids, we don't have any chromatids in this place, so we're going to say there are none. And remember, chromatids, if you recall from maybe your instructor or online resources, they're called chromatids only when you have two DNA strands connected together at a central location, which is that centromere, and I'll talk about that a little bit later. Let's do another example. Here we have two DNA strands, okay, and we have here two chromosomes. All right, the reason why is because we have two separate strands of DNA, therefore two separate entities of DNA, and so therefore two chromosomes. We still don't have chromatids here because in order for them to be called chromatids, they would have to be attached at the center. Let's do a third example before we continue on. How about this one? All right, so how many DNA strands do we have here? We still have two DNA strands. So I'm still going to say two DNA strands. But how many chromosomes do we have? Only one. And the reason why, because a chromosome is an entity of DNA, and as you can see here, we have one entity. These two DNA strands are connected, but it's one piece, okay? One piece, one solid piece, and so therefore we're going to say this is one chromosome. Now we have two chromatids. And the reason why they're chromatids is because these two DNA strands are replicates of each other and they are connected there at the centromere. So we're going to say this here is a chromatid and this whole thing here is a chromatid. Okay, this whole piece here. Okay, this one. Let's do one more example. How about this case right over here? So how many DNA strands do we have? I'm going to color code this one here. We have here 1, 2, 3, 4. So we have 4 DNA strands. How many chromosomes do we have? Well, we've got one entity, two entity. This one's connected. So this is 3 entities. So 3 chromosomes. But how many chromatids do we have? Well, let's count them. That's not a chromatid because it's not connected at something called the centromere. That's not it. But these two are because they are connected here. So we've got one chromatid here and a second one there. So we've got 2 chromatids. Okay? So that's just a quick rundown on how to count the number of DNA molecules, chromosomes, and chromatids. All right, let's go ahead and get straight to mitosis. All right. Now, just to refresh your memory, mitosis in the mitotic phase, right? In the mitotic phase, there are 4 phases of mitosis. There are 5 technically, depending on the textbook that you're using, but I'm going to focus on these main 4. First one we're going to say is prophase. Second one is metaphase. Third one is anaphase. And the fourth one is telophase. And I like to link cytokinesis with it too. Depending on the textbook, they might link it. All right. Hope that you're taking these notes down. Okay? So let's go ahead and get started with our drawings. I'm going to go ahead and erase the board. Start here. Now, before we begin with mitosis, I'm going to tell you how many chromosomes are in this cell. Okay? The number of chromosomes, I'm going to say, in this example, is going to be 4. I'm going to use 4. Now, you might see this number 4 written as 2n equals 4. No problem. Okay? But just keep that in mind that some of your instructors or whatever may use 2n equals 4 instead of just saying number of chromosomes is 4. All right? So just keep that in mind. So the first phase of mitosis, if you remember, is prophase. I'm going to draw a line here to separate my work. We're going to go ahead and draw prophase. So let's start. Prophase begins with a cell membrane. And so I'm going to go ahead and draw a circle on my board. That is a cell. And I'm going to go ahead and label this prophase. We have here inside of that circle a nuclear envelope. So it's like a circle inside of a bigger circle. Okay? But this inner circle here, I'm going to draw dotted in there. Because what's going on is a nuclear envelope is breaking down. We have these structures. Okay? And these are called centrioles and spindle fibers. And then inside of this cell, I'm going to go ahead and draw the number of chromosomes, which we have here is 4. Now, just to let you guys know that before prophase, we did have something known as G1, S, and G2. We're not worried about that component of the cell cycle. I'm just going to jump straight on into prophase. Okay? So you might need to do a little review on how the chromosomes look in G1, S, and G2 from your instructor's notes, your textbook, etc. In this case here, we've got 4 chromosomes. So I'm going to go ahead and draw them. Watch how I draw it. Okay? There's 1. There's 2. There's 3. There's 4. Now in the diagrams that I'm drawing, I'm not using different colors to represent the father's DNA and then the mother's DNA. I'm just strictly drawing chromosomes, okay, in the same color just to keep it simple and quick. I'll let your instructor change those colors to match the mother's or the father's, but this one is just to show you how it works. Okay, and once again, these are chromosomes. All right. We also have here, this should be labeled as the nuclear envelope. Okay. So that should be prophase. Let's move on to the next step. Metaphase. I'm going to draw another circle. And now in this step, the nuclear envelope has already broken down. Those little centrioles and spindle fibers that we drew earlier, they've migrated to opposite poles of the cell. Okay? And the chromosomes have now lined up at the center right here. This is an imaginary line. It doesn't exist in the cell. It's just an imaginary line to help you see it. But all the chromosomes, as you can see, now they're lined up at the center. And notice how I'm drawing these chromosomes. I'm not drawing X's, because every time I draw X's, I get a lot of students that get confused with X's. But when I draw them like this, people get them. Okay? So now we have here, I'm going to the centriole, and then I'm going to draw a spindle fiber coming out, and it has to touch that centromere. That's absolutely key. Same thing here. It's got to touch that centromere. Got to touch that centromere. Okay? And same thing for the other side. It's got to touch that centromere. All right? Now, we also have they call non-kinetochore connecting spindle fibers, but I'm not going to worry too much about that. Okay? This imaginary line that we saw earlier is called the metaphase plate or equatorial line. Depends on which book you're using. All right, let's move on to the next one. After metaphase, we've got anaphase. So I'm going to draw the arrow to go down this way. And in anaphase, as you recall, the sister chromatids are separating. So I like to draw the sister chromatids first, and it's just like when you're tugging on a piece of a rope or something like that. You get this force and this bend in the shape. You can see here. Okay, so it's not ripping them apart, but it's pulling them, wheeling them apart, just like so. Let's go ahead and draw our centrioles. They're still on the opposite poles, and they're still connected to that center region right over here. Okay. Make sure that they're touching. Okay. And of course, we have our kinetochore spindle fibers. That's anaphase. And now we have the last one, which is telophase. Now, each instructor might have their own unique way to draw a telophase, but in my class, I draw it like this. I like to draw them as if the cells are ready to split. So you can see there. In this case, these 4 now chromosomes are on one side and the 4 are on the other side. Okay, that nuclear envelope is coming back. It's reforming. There. Those spindle fibers and centrioles are now kind of breaking down. They're not needed anymore. And we have this structure here in the center that I want to draw. Okay, that's known as the cleavage furrow. I don't have enough colors to go around to label this, but here we go. We're going to call this the cleavage furrow. Okay? And once again, this is telophase. Cytokinesis. I just want you to know that cytokinesis is the actual splitting of these 2 cells, but I usually, in my class, I just label it like that. Okay, just for the drawing's sake. Okay, so we have here a cleavage furrow. We've got here our nuclear envelope that's reforming, and we have our chromosomes that are in here. Now, you're probably wondering, how do I count the number of chromosomes, chromatids, and DNA molecules? Let's go ahead and do it. Excuse me. So as you can see here, let's go ahead and go to prophase first. So I'm going to say DNA, chromosome, chromatid. So how many DNA molecules do we have here? Well, in the nucleus, I have DNA here. There's 1, 2, 3, 4, 5, 6, 7, 8. So let me count that one more time. DNA molecules. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 of them there. Now the number of chromosomes. Well, how many entities do I see? I see 1, 2, 3, 4. And now how many sister chromatids do I have? 1, 2, 3, 4, 5, 6, 7, 8. I have 8 sister chromatids. Okay? Now if you want to, you could say how many centromeres do you have? There's 1 centromere, 2, 3, 4, and then multiply that by 2. So 2 times 4 gives you 8 sister chromatids. Okay? What about in metaphase? Over here. I'm not going to write this. I'm going to assume that you know that. Okay? How about in metaphase? How many DNA molecules? 1, 2, 3, 4, 5, 6, 7, 8. How many chromosomes do we have? Well, 1, 2, 3, 4. And then how many sister chromatids do we have? Well, 1, 2, 3, 4, 5, 6, 7, 8. Okay. How about an anaphase? This is the tricky one here. How many DNA molecules do we have? Well, 1, 2, 3, 4, 5, 6, 7, 8. Still 8. How many chromosomes do we have? Is it 4? No. It's not 4. It is 1, 2, 3, 4, 5, 6, 7, 8. Okay? And the reason why there's 8, because you have 8 entities of DNA. So there's 1 entity, 2 entities, 3, 4, 5, 6, 7, 8. Okay? This is a tricky one. And how many chromatids do we have? Well, we don't have a centromere. Okay, they're not connected. They're not connected at the centromere, so it's got to be 0. Okay? And lastly, for mitosis, right over here on telophase and cytokinesis, how many DNA molecules do we have? This is tricky now. We have 1, 2, 3, 4. So it's 4. Now, it's per nucleus. Per nucleus in here, or in here. 4 in here. How many chromosomes do we have? 1, 2, 3, 4. And how many chromatids do we have? None. Okay? All right. So hopefully this diagram will kind of clarify how to draw mitosis. And again, we are talking about 4 chromosomes. All right, I hope that you guys found that mitosis tutorial, that virtual lab, useful. Once again, if you don't quite understand it, I strongly recommend that you go back and move that slider to the back or whichever direction this video is going to turn out, and go ahead and go through the video one more time and drawing each of those steps along with me. All right, it really helps, and I would strongly recommend that you have colored pencils with you too, and draw them in different colors if it helps you understand those concepts. Okay, and then so anyway, hopefully that you found the video helpful and it's useful for you in your understanding of mitosis. So until the next time, see you around.